Today we will discuss about pancreatic cancer cells and how this pancreatic cancer cells functioning can be disrupted by disrupting the intercellular transport that helps in maintaining the functioning of the pancreatic cancer cells. And one of the intercellular transport or intercellular transport that this paper discusses about is this RPS18. So what this RPS18 does is RPS18 is transported using exosomes in the tumor microenvironment, and it helps in maintaining the lipid metabolism of the pancreatic cancer cells. The researchers have found out that the immune cells, that is monocytes, which are differentiated either into M1 macrophage or M2 macrophage, and we have discussed this thing in one of our previous videos that M2 macrophage has the pro-tumorigenic properties, whereas M1 macrophage has the anti-tumorigenic properties. So, however, how this M1 macrophage acts as an anti-tumor is not been discussed in detail. So, one of the aspect of this anti-tumor properties is provided in this uh, brief overview. So, it starts with this IRF7. So IRF7, what it does is it will reduce the production of this RPS18. This RPS18 reduction will cause less amount of RPS18 will transport via exosome into the pancreatic cancer cells, and this less RPS18 will transport, then it will cause less productions of this ILF3, and ILF3 is related to the ILF3 is related to the this lipid metabolism of the pancreatic cancer cells, and once this disruption in the lipid metabolism occurs with time, it will cause disruptions in the normal disruptions in this pancreatic cancer cell functioning and even trigger apoptosis. We will discuss the experiments that they have done to come to this conclusion. And but for today's video we will just focus on their overall objective that they have presented in the paper. So when the lipid metabolisms get disrupted, then also what will happen is that it will trigger apoptosis within the pancreatic cancer cells, and this could be, and based on their study, this RPS18 or this intercellular transport of this RPS18 could be one of the potential target for the therapeutic approach, so that can be used to reduce the pancreatic cancer cells within the patients. So, and these things they have observed in the tumor microenvironment where M1 macrophages are present compared. However, in this figure they have not mentioned whether this kind of intercellular transport also occurs from other immune cells or the other cells that are present in the tumor microenvironment. Uh, so once this lipid metabolism get disrupted, it will cause this kind of changes, that is viability of the pancreatic cancer cells get reduced, migration properties. The pancreatic cancer cells are known to be very aggressive cancers that migrates across different part of the body, and the proliferations also reduce, so the growth rate of the pancreatic cancer cells will get reduced, and the deletions, invasions also reduced, so that the ability of the pancreatic cancer cells to spread across the neighboring cells will also be reduced, and the apoptosis is increased, so it will cause the death, or it will trigger the death of the pancreatic cancer cells, and thereby reduction in the pancreatic cancers within the patients. We will discuss the experimental details and their results in the subsequent videos. So however, if you need to go through that paper that presented this hypothesis, or that presented this phenomena, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.